and this is my presentation on Chapter 9. So first I'm going to talk about the essential nutrients. So microorganisms, they require nutrients to sustain their life, and essential nutrients just means what they cannot make on their own. So there are 6 key elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, and these are known as CHOMPs, and these are the nutrients that are crucial for most macromolecules. So nitrogen, it's essential for nucleic acids, proteins, and phosphorus for ATP. And then without these, the microorganism would not grow or reproduce. It would just basically just be there. So then there's also other elements that microbes require, but in much smaller amounts. So like potassium, calcium, iron, magnesium, chlorine, and then there's also trace metals. These are only needed in super tiny amounts, but they act as cofactors for enzymes to help carry out catalytic functions correctly. So now I'm going to talk about macronutrients and micronutrients. So nutrients is divided into these two categories. Micronutrients, these are the ones that they need in bigger quantities. So they're like proteins, carbohydrates, lipids, nucleic acids, and they include the CHOMPs that we just saw, and they play a huge role in cell metabolism. And then there's also micronutrients. So these, like we said before, are needed in much smaller amounts, but they're still very essential. So nickel, zinc, copper, these act as cofactors for enzymes and help catalyze reactions, and they also play a role in protein stability. And even though they're very small, they would completely stop metabolism without them. So now I'm going to talk about the carbon and energy sources. So microbes, they differentiate how they obtain their energy and carbon. So heterotrophs, they rely on organic molecules like sugar and proteins and lipids for their carbon source. And then autotrophs, they usually convert inorganic carbon from like carbon dioxide. They convert those into organic molecules. Then there's also microbes that only obtain their energy from light, like phototrophs is what they're called, while there's others that get their energy from chemical reactions, and these are labeled chemotrophs. And then there's also combined categories at the bottom: phototrophs, autotrophs, photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. So humans are chemoheterotrophs. They get their energy from chemicals and they depend on organic carbon. But there's also chemoautotrophs. These are like bacteria living in extreme environments. And photoautotrophs, these are like plants, algae. They use sunlight and carbon dioxide to live. And then I'm going to talk about the elements in CHOMPs. So they each play a different role. So nitrogen, it's essential for amino acids, nucleic acids, ATP, but they can't get their nitrogen gas directly from the atmosphere. They have to get it from specialized bacteria or on nitrogen that's already been converted into what it needs, like ammonium nitrate or nitrite. And then there is oxygen. So oxygen, you think it's essential for all organisms, but it can actually be toxic, and many organisms will use enzymes like catalase to like detoxify it, basically. And oxygen is also important for the production of proteins, lipids, carbs. And then we have hydrogen. So hydrogen plays a huge role in maintaining the pH balance, and hydrogen bonding also is a very strong form of bonding, so it helps stable the molecules through hydrogen bonding. And then it also plays a role in oxidation and reduction reactions. And then we have phosphorus. So phosphorus is most commonly found as phosphate. It's an important structural component. It helps with nucleic acids, ATP, phospholipids. And then we have sulfur. So sulfur is usually found in minerals and in amino acids like cytosine. And this helps form disulfide bonds. And disulfide bonds are used to help stabilize protein structures. So now we're going to talk about saprobes and parasites. So there's many different strategies they use to get their nutrients, microbes do. So saprobes, they thrive on decaying material or dead animals, leaves that have fallen. So that's where they get their nutrients. So they use enzymes to secrete the external organic thing and get the nutrients and recycle them back into their self and use these to live. So then we have parasites. So parasites, these are different because they use a living host and can actually cause harm to their host while feeding off of it. So viruses and some bacteria can fall into the category of parasites when they hurt their host. So we also have facultative parasites. These usually they live as saprobes like freely, but if they're able to, they'll shift and can turn into a parasite and take advantage of their host. So then we have opportunistic pathogens. These usually don't cause harm to their host, and they usually infect people that have a weak immune system. So I'm going to talk about the different ways that cells interact with their environment and transport stuff. So we have diffusion and osmosis. So diffusion, these are how molecules will naturally move inside the cell. They move from areas of high concentration to a low concentration until some equilibrium is reached. And then osmosis, this is how water moves. So it's a specific type of diffusion. Water moves across a selectively permeable membrane, and there's different environments that must happen. So we have, it just depends on the type of solution that's in the cell. So there's isotonic, and this is when the water moves in and out equally, so the cell is stable. But in a hypotonic environment, water will rush into the cell and it can swell or even burst. And then there's hypertonic. So hypertonic is when water will leave the cell and it'll cause it to shrink or even become plasmolized. So now I'm going to talk about the different types of transport, passive and active. So passive transport does not require energy at all. So simple diffusion, this will allow small molecules to just go through the lipid bilayer. And then there is facilitated diffusion for larger polar molecules. And they will use protein channels or carriers, and they help make this movement possible because of the protein gradient. So the protein gradient provides it with enough energy to... Oh wait, I think the protein gradient's for the next. Okay. Then there is active transport. So active transport requires energy. So usually in the form of ATP. And I'm going back. It wasn't the protein gradient. Facilitated diffusion is powered by the concentration gradient. Okay. Active transport pumps against the protein gradient. It uses ATP, and it's essential because it might have toxins come into the cell, and it'll want to get rid of them quick. So then it'll use its energy to get the toxins out of the cell quickly. And then there's a special category called group translocation. This will modify the substances as it comes into the cell. So like glucose to the glucose 6-phosphate. So now I'm going to talk about how the more complicated ways of transporting substances. So we have endocytosis. This is when the cell will fold inward and it will enclose onto whatever is trying to come in and bring it into the cell. And then there's just the opposite process, exocytosis. This wants to push stuff out of the cell, it'll expel it, and this is good for waste removal. Then there is like these special forms of endocytosis called phagocytosis and pinocytosis. So phagocytosis, this is like when the cell eats something. So like macrophages, it wants to engulf these large particles. And then there is pinocytosis. This is cell drinking. So it wants to take in the extracellular fluid or any dissolved nutrients in by small vesicles. And that's the two different types, pinocytosis and phagocytosis. So now I'm going to talk about the different factors and that alter microbial growth. So microbes, they live in many different places, whether it's like extreme hot environments like volcanoes or like at the bottom of oceans. So their survival is dependent on their ability to adapt to these external conditions like pH, temperature, or oxygen. And microbes will be divided into different categories depending on what their tolerance is. Oh, and tolerance. Tolerance is when the conditions aren't just right, so they will just go into like hibernation. Basically, they won't grow and they'll just remain the same until the conditions improve. So now we're going to talk about temperature and its effect on microbial growth. So temperature is divided into, well, each microbe will have a cardinal temperature. So there's minimum, optimum, and maximum. And if the temperature is outside of their range, the activity of their enzymes or stability of their DNA, it can go south really quick and the organism can die. So there's also classifications for their different temperature range. So psychrophiles, these thrive in super cold environments like polar ice or the bottom of the ocean. And then we have psychotolerant. These are organisms, they live in like cold but not warm. They thrive in like refrigerated temperature, and they play a role in food safety because they can survive in those environments. Then we have mesophiles. These are the most relevant group because they survive in the same temperature that we humans survive in. And then we lastly have thermophiles. So these survive in hot temperatures like hot springs or volcanoes. So now we're going to talk about oxygen. And we said before, it's usually required for life, but it can also be potentially lethal. And oxygen, it participates in metabolism, but it also produces radicals as a byproduct, which can damage lipids and proteins and DNA. So organisms that survive in environments with lots of oxygen, they produce catalase and superoxide dismutase, and these basically take care of the oxygen problem. They detoxify it. And microbes, they're classified into groups based on their oxygen requirements. So some require it completely and some others can tolerate it and some don't need it at all. So the different oxygen categories, we have obligate aerobes. So these thrive in oxygen-rich environments. And then we have obligate anaerobes. So these will be killed when exposed to oxygen. So then we have the facultative anaerobes. These are important because they're very flexible. They can survive inside and outside the body. So they don't need oxygen, but they can survive with it. And then we have aerotolerant species. These are like another that don't use oxygen, but they can survive it. So there's other factors like pH, and there's acidophiles and alkalophiles. So acidophiles can thrive in extreme acidic environments like lakes, and alkaphiles can live in extremely basic environments. Then we have osmotic pressure as another factor. So